this would be associated with, for example, a reduction in the acceleration of the grey matter that we thought was more likely to be the case in children with conduct problems. I'm going to just show you some of the results. So this slide here highlights the fact that the dedicated team of people working on the study with me over the time that we've been doing it managed to recruit 80 kids with conduct problems and 50 control children. I think we're getting up to 100 now. And they were scanned at 2 time points: before their parents had been told how to be or taught how to be better parents, and then after their parents had gone through the parent training. Well, one thing we found was that the conduct problems between the 2 time points reduced.
Sorry to bother you. Would you have the room booked at 12:30?
Okay. What do you want me to do? 5 minutes?
Say again?
5 minutes? I'm
yeah,
finishing a lecture.
Say, yeah. That would be great. Okay, thanks. Thank you.
Well, that's never happened before. I'll try and go as quickly as I can. So we had a reduction in the conduct problems. What was interesting, though, was that when we were looking at the effects of callous unemotional traits, we found that in the children who had low callous unemotional traits, if the parenting improved between the 2 time points, we got a significant reduction in their conduct scores. Whereas in those children who had high callous unemotional traits, even if the parents' parenting style was changing, there was no significant difference between the 2 time points. So p-value is 0.504. So even though parenting may be helpful, it may not be helpful in a particular group who have these more severe callous unemotional traits. So the next question is, well, what do we do about them? And that's what we're thinking about at the moment. I'm just going to go over this slide very quickly, but basically what we also found is in the children who had reduced conduct scores, so these are basically the conduct disordered kids, these are the kids that basically got better over the 2 time points. We had reduced acceleration of the grey matter in regions like the amygdala, particularly the amygdala, compared to those kids that persisted to have conduct problems and didn't respond to the parenting training. So we can't say exactly that the parenting training is slowing down this brain acceleration, but there is some evidence perhaps that parenting and the behavioural changes that we're seeing are correlated with changes that we can see in the brain, which I think is really exciting. It's really exciting for a number of reasons, one being that people, I guess, see things like parent training and therapy as being a bit sort of fluffy and not necessarily hard science, but when one can actually see changes happening in the brain correlating with changes in this behaviour, that does put, I think, a different slant on things. And hopefully at a time where these parenting programs are being cut by the government, something that will encourage the government perhaps to put more money into these sorts of things, but perhaps directing them towards groups that are most likely to respond. Now, I don't know if I've got time to go over this in too much detail, so I'm going to fly through it. This slide here highlights the fact that if you're a mum with, or if you're born to a mum with depression, you've got a much higher chance of having depression later on in life. And that if you do have depression, one of the other things that we notice is that you're more likely to focus on negative things rather than positive things. So there's a bias in people who are depressed or predisposed to depression to take more attention to something that's negative than positive. And in babies that have been born to, children that have been born to, yeah, I've already been told, but I'm going as quickly as I can.
Okay, we have a talk, a 1-hour talk, and our guest speaker just arrived.
Okay.
Thank you.
I may have to come and give the rest of the talk. There's only about 5 slides at the end of another thing. Yeah. Sorry about that.
can all stay for this one if you want. Autism and reproducibility and proper science.
Did you just say proper science?
Yeah, proper science.
Were you on a mic, or is this as loud as it gets? Oh, does that project back? You can hear me? Am I mic'd? Or is
No,
that-
I think, can everybody hear?
Those
Is it
people at the
you
back
all-
probably not going to hear me.
Oh, don't worry about those at the back. They're just, they're the heavies, if anything. It's like that. Okay, well, we'll make a start anyway.
Okay, fair.
Yeah, so we'll make
So
a
don't run
start
too far from
anyway.
that. Yeah. Okay.
So, this is the RIOT Science talk, not the whatever that was before. So, we're joined today by Dr Hendry, who's come all the way from Oxford today, so we're very appreciative of that jaunt. And you've escaped the extinction rebellion, which apparently kicked off last night in Town. Anyway, so you did your PhD at King's.
I did.
And it was in executive functions and ASD and ADHD as well, development of executive functions. And you're continuing that work at Oxford as well. And I need to thank, where is Amy? Is Amy in here? Oh, there. I need to thank Amy because when I originally came up with the idea for a series of talks about the difficulties that ECRs face in reproducibility and the sort of the crying matches that happen, and suggested your name. And
She's like, she cries a lot, yeah.
And so Amy gave a talk a couple of weeks ago that sort of went through all the problems that she's had, and we'll have the angle from ASD today. So thank you again for coming in and talking about this that needs more airing, I think. So I'll stop talking because we're running late as it is, and I'll let you get on with it. So
Okay, great, thank you. All right, so the basic content of my talk is all about this idea of understanding early development of attentional control. But what I'm going to do is try and kind of bring out reflections on the OSFE, Open Sciencey elements of it in terms of how I've attempted a pre-registered self-replication and some of the strengths and difficulties of that approach. So I'll start by outlining why I think it's interesting and important to investigate the early development of attentional control in infants with an elevated likelihood of autism. And then I'll explain how we can use data-driven approaches to better understand early development. I'll talk about the results from a discovery study that I did as part of my PhD, and then I'll explain how and why I went about pre-registering a self-replication and kind of draw out some general study findings and lessons learned from that process. So I'll start the way every autism talk ever starts, with the core diagnostic criteria for autism spectrum disorder, which are calculated as social communication atypicalities and rigid and repetitive behaviours. And that's nice and clear-cut, except it kind of belies a huge range of heterogeneity in that individuals vary both in the extent of autism-related traits within these domains and also in their intellectual ability, and importantly in their ability to function independently in day-to-day life. And we know that autism is highly heritable. Family studies and twin studies have shown us this. And yet attempts to find a single genetic account of autism have largely failed. And we think in a great extent this is due to the heterogeneity of autism. And that's made even more complicated by the fact that autism co-occurs with a lot of things, and one of the things it frequently co-occurs with is ADHD. So around 30 to 80% of people with autism also meet criteria for ADHD, and around 20 to 50% of people with ADHD meet criteria for autism. So trying to kind of unpack that and the overlap between is very tricky. So one way forward is to take a different approach, to take the approach advocated by the research domain criteria. And that suggests that we might want to look past diagnostic boundaries and actually think about underlying transdiagnostic systems. So kind of zooming in on one particular mechanism or system that might vary across populations and contribute to multiple developmental conditions. And these transdiagnostic systems or intermediate phenotypes may well have a clearer genetic profile, which might help us better understand the etiology of that particular facet of autism and ADHD. So that's kind of the basic premise. And the transdiagnostic profile that I was interested in is attentional control. And there's kind of a strong justification for that. So a lot of the overlap between autism and ADHD may well share common antecedents of genetic origin. And there's a kind of a big literature suggesting that that relates to difficulties with attentional control. So that's really interesting in and of itself, I think, to try and understand the etiology of these conditions. But I think it's also really, really important that we remember that we're talking about people here. We're being funded by the taxpayer and by charities to try and make a positive impact on people's lives. So one thing that I'm really interested in understanding is, well, what do these difficulties mean for real-world outcomes? How does variation in the development of attentional control support or restrict the ability to function independently? How might we be able to spot early disruption in attentional development and intervene to support optimum outcomes? And who might need those extra supports, and what might those extra supports be like? So that's kind of the underlying thing motivating my interest in the question. Before I go any further, I'll just explain what I mean by adaptive functioning. So by that I mean the ability to cope with everyday situations at an age-appropriate level. So that's going to vary across development. And we know that autistic children with co-occurring ADHD seem to have extra difficulties with adaptive functioning, but very few people have considered this from an underlying transdiagnostic phenotype approach. So is it attention difficulties per se that are driving these extra difficulties? So there's been not very much research into that question, and there's been even less research trying to understand that developmentally. And I think it's really, really important to think developmentally, because these are developmental conditions and because humans are messy. So we are the result of numerous interactions and feedback loops that kind of influence us across development and have kind of cascading effects. And so in the autistic context, attention difficulties may well be a consequence of atypical, perhaps social interaction early in development. And so if we want to understand, well, is attention actually the phenotype, the transdiagnostic system driving those difficulties, we have to kind of unpick some of those feedback loops, and we have to look earlier in development. And when I say earlier in development, I mean really, really early. So the ability to control one's attention emerges in the first year of life. So from around 9 months of age, infants are able to endogenously control their own attention. Prior to that, they're slightly at the mercy of the environment. You know, a stimulus comes in and they're redirecting their attention or fixating their attention accordingly. But from 9 months, they begin to have endogenous control, and that control acts as a foundation for the development of many more complex, higher-order skills. So it's incredibly important to later development. So that's why we need to understand early attention development. But I'm talking about early attention development in autism. How do we do that prior to the age of 3? Because conventionally, autism is diagnosed in the community from around 5, even in research settings, generally from around 3, it's considered a robust diagnosis. So how are we finding these autistic babies at age 1? Well, that's challenging if we just took a random sample. So the community prevalence rate is around 1%. So if I sampled 100 babies from the local hospital, I'm only going to find one child who's going to be given a diagnosis of autism. But as I mentioned, autism is highly heritable. So if I specifically sample infants with an older brother or sister or a parent with a diagnosis of autism, that puts them at an elevated likelihood, around about a 20% chance of developing autism. And a further 30% would be expected to show subclinical traits. So elevated autism traits, not necessarily diagnostically relevant, but certainly relevant at the trait level. And so that's what we do in the BASIS project that I'm a part of, where we take a longitudinal infant sibling design. So we follow up these babies who are at elevated likelihood of autism, and we capture that at critical points in development. And for me, this is around 10 months, 15 months, 25 months, and 3 years. And at age 3, we can use a battery of kind of clinical measures to ascertain whether they do in fact have autism or not. So that's the basic premise of the design. But of course, then I've got to be thinking about, well, what am I doing at these key points in development? How am I understanding attention development? And we can do that in many ways. We could use eye tracking, we could use behavioural tasks, but the measure that I'm talking about today is parent report. And I think parent report is particularly useful for a number of reasons. Firstly, it's ecologically valid. We're talking about behaviours that are relevant in day-to-day life. Secondly, because it's given to us by parents who are with the babies all the time, it's broad and it gives deep insight across time and across context. And that's really important when thinking about attention in particular, because a baby in a calm, very quiet lab environment might find it very easy to focus their attention, and that might not correspond to their actual behaviour in day-to-day life in the kind of chaotic real world. So it's important from that respect, but it's also important just from a practical respect. Parent report is basically cheap. It's easy to collect large samples with parent report in a relatively standardised way. And that might be relevant when we're talking about, well, what can we do with this kind of research? So I'm interested in informing screening work, and you're never going to be able to do mass population level screens with intensive eye tracking or behavioural tasks. Whereas with a parent report, that's suitable for geographically dispersed populations and low resource nations. So the kind of parent report that measures that I use are based on the temperament literature, and there's 2 dominant questionnaires, both from the same group. For infants, we use the Infant Behaviour Questionnaire, and for toddlers, we use the Early Childhood Behaviour Questionnaire, and they're designed to follow on from each other. So in infancy, we particularly use the Duration of Orienting Scale, and that asks us things like, during the last week, how often did your baby stare at a mobile for 5 minutes or longer, play with one toy or object for 5 to 10 minutes, look at pictures and books for 5 minutes or longer? And that actually does give a lot of variation, even at 9 months. And then by toddlerhood, toddlers are capable of a kind of bigger, wider repertoire of behaviours, so we can ask more about attentional focus, so we can again ask about how they're playing with objects for long durations of time, but also their distractibility, and their ability to kind of persist with slightly challenging tasks. And on then kind of the other side, we can also ask about attention shifting. So how well are they able to redirect their own attention between target and target? So because these measures have been around for 20, 30 years, we have a lot of evidence to support the fact that these are robust measures that capture something meaningful. We know that they're sensitive to developmental progression, so we see increases in attentional control scores from 9 months right the way through to early childhood. Yet within that developmental progression, we can capture fairly stable individual differences. And we see moderate to large correlations across spans of 6 to 18 months, and that's really quite rare in my work. They're also, importantly, they're predictive of later behavioural difficulties. So Gartstein and colleagues showed that low parent-reported attentional focus in toddlerhood is associated with elevated internalising and externalising problems in the preschool years. And that was just in a typically developing sample. So then what do we know about parent-reported attentional control in autism? Well, a fair amount. We know from age 2 years, amongst children who've already had a diagnosis of autism, there's plenty of evidence for difficulties with attentional focus and attention shifting. That's a fairly robust literature. But it gets a bit more kind of messy when we look younger in development. So Garin and colleagues found that there was evidence for difficulties with attentional focus and attention shifting amongst these infants with a family history who are at elevated likelihood of autism generally, and specifically amongst those infants who go on to receive a diagnosis of autism. And yet another paper from our group showed that there are no evidence for group differences, either in this kind of elevated likelihood or subset who go on to develop autism at 14 or 24 months. And then in terms of ADHD, Frick and colleagues have found recently that in a typically developing sample, individual differences at 10 months of age in a kind of composite attention regulatory measure are predictive of ADHD-related behaviours at age 3, so that's quite promising. And yet, Miller and colleagues found that kind of broad parent-reported concerns, which included poor attention, were picking up difficulties at age 2 and 3 years, but not prior to that. And recently, Lizzie Shepherd from our group showed that there is no evidence amongst infants with a familial history of autism, so who are at elevated likelihood of autism, but go on to show high ADHD traits. Those children weren't scoring low on attentional focus at 7, 14 or 24 months. So it's a slightly cloudy literature, maybe it doesn't look that promising, but there are a number of reasons for this. So it could be that actually, development diverges from age 2 years. So there's something kicking in about attention from toddlerhood that starts to drive these difficulties. Could be that. It could be that our parent report measures of attention in infancy aren't sensitive enough to detect genuine differences, possibly. But I would argue that maybe it's more about the latter 2 points. Maybe we shouldn't be looking at differences at a single point in time in infancy, but rather at differences in development, because these are developmental conditions. And maybe genuine differences amongst some infants with a familial history of autism who are at elevated likelihood are masked by heterogeneity within the groups. So that got me thinking, well, what if we don't start from the assumption that there are these kind of inherent differences between these elevated likelihood infants and these typical likelihood infants at a single point in time? What if we take a more developmental approach, looking at change over time? And what if we identify our groups on the basis of the data and not our recruitment strategy? So I persuaded my supervisor to kind of let me out a whole bunch of parent report data that had been collected over many years from the British Autism Study of Infant Siblings. And because, over time we finessed our protocols, the age bands were a bit drifty, so I kind of cut out babies who didn't fall into nice clear camps because I wanted to be sure that I was comparing like to like. So one side just collated infants who had data at 9 months, 15 months and 25 months. I had 212 infants with elevated likelihood of autism and 82 typical likelihood infants. And the whole idea here was to use a latent class analysis approach. And what we're aiming to do with this is to recover phenotypically homogeneous subpopulations from within a heterogeneous sample. Or in other words, to take a big messy group of infants who are varied in many, many different respects and kind of use the data, use the model to divide them up into subgroups, to sort through them until we found groups that are like each other on a particular domain of interest. And of course, the domain of interest for me is attentional control. And I ran that model without reference to likelihood group, without reference to diagnostic group, and without any input on symptom measures. So this is a completely blind model. It's only using the parent report data. And the way it works when you're kind of trying to come up with the right model is that you keep adding the number of classes. So I put in the duration of orienting scores at 9 and 15 months, and the attentional focus scores at 25 months, and attention shifting at 25 months. So this isn't a latent growth model. We're not looking at trajectories per se here, because attention focus and attention shifting are at the same time point. We put in all that data kind of in a bottom-up way, and we tell the model, well, keep adding classes until I'm no longer getting you better. And so we use the model fit statistics to compare that. And in this case, the model stopped improving at 4 classes. So what do these classes look like? Well, we can see, I think, some quite clear trajectories, but I'm just going to orient you to that a little bit more. So the first profile is, very clearly high attentional scores across the board. The second, and that's 17% of our sample. The second group show initially quite low attentional control scores at 9 and 15 months, and then they seem to just kind of get it. They end up looking pretty similar to the high group. But here's where the bulk of the kids are. 60% of our children get classified to this, what we call normative profile, because that's just where most people are. And they're showing, fairly medium scores, and some developmental improvement, which is what you would expect based on the typical development literature. So that's all very well, but the group that I'm particularly interested in is this group, what I'm describing as a plateaued profile. So they start with kind of middling scores just like the normative class, but then they fail to show the expected developmental increases. And this is characteristic of 7% of my population. So then I kind of was like, well, what does this mean? Is that a genuine class that actually impacts on their attentional focus in the real world? So I was able to then see if that would predict their attention scores at age 3. So outside of my latent class analysis model, did it relate to their 3-year attentional focus? And what we find is that the normative class have a mean attentional focus score of 4.4, and the plateau group have significantly lower attentional focus at age 3. So that gives me some confidence that what we're capturing here is atypical development of attentional control. Okay, great. But what I'm really interested in seeing is, well, what does that matter? Does that matter to their autism traits, their ADHD traits, and to their adaptive functioning? So then I kind of predict, again, outside of the latent class model, predict these kind of distal outcome variables. So the first variable I'm looking at is the SRS score. So that's our measure of autism traits, where a high score is lots of autism traits, elevated autism traits, and low score is pretty much typical development. So you can see our normative class has a score of 22.5, whereas our plateaued attention development class is scoring at nearly 95, which is well within the range of clinical concern for autism. Then in terms of ADHD, and that of course is statistically significant. In terms of our ADHD scores, here I'm using the CBCL, so this is only looking at trait levels. 3 years is way too early to be diagnosing ADHD robustly. But we can see that the normative class is scoring around 4.5, and the plateaued attention development class is scoring significantly higher. Not yet within the realms of clinical concern, but they're 3. So it's really early days for ADHD traits to be emerging. And then in terms of adaptive functioning, so here I use the Vineland, and that's normed to a score of 100 with a standard deviation of 15. So you can see the normative class are pretty much bang on the norm, but the plateaued attention development class have significantly lower adaptive functioning. So I was really excited by that. I thought I'd captured something interesting and new about the data, but my supervisor was not so convinced. And he kind of flagged up a number of limitations in the way that all good supervisors should. So the first issue was my sample sizes. So I started off with this really lovely hundreds of infants, and then as soon as you start identifying subgroups, I was down to only 20 infants of particular interest to me. So that was a problem. The next problem is, well, yeah, I've accurately described the infants that I've got, but maybe that doesn't generalise to anyone else, so really, who cares? And then the third question is, I've convinced myself that that's what I was expecting all along, but I didn't pre-register it. I've kind of post hoc rationalised this nice, neat explanation. And as we all know, the only way to solve that is to pre-register your hypotheses. So that's what I aimed to do. So my next mission was to find another large sample of elevated likelihood and typical likelihood infants with suitable data to check whether I get similar classes. They didn't have to be identical because this is a new sample, so you're going to expect the infants to be slightly different, but do we get a similar kind of profile of atypical development in there somewhere? And then most importantly, do I get the same findings that atypical development of attention is predictive of elevated autism and ADHD traits and of weaker adaptive functioning? And that's the bit that I was really keen to pre-register. And then I also kind of wanted to look just in a more exploratory way about whether elevated likelihood and typical likelihood groups would differ, and whether children with a diagnosis of autism or not would differ. And because my sample sizes were really quite small in my discovery set, I didn't have a clear answer on what I would expect there. So in my pre-registration, I just kept that open. I was able to just say, well, this is what I plan to look at, but I'm not going to say what I expect to find. So, how did that go? Okay, so my first challenge was to find another large sample. So I guess my first kind of positive comment on that is that when you've already got an interesting exploratory finding to build on, it's relatively easy to get people to share with you their data so that you can then do the big paper using their data and they get named and everyone's happy. So I'm very pro discovery samples, finding something interesting, and then doing it with someone else's data later. And I just had a really, really positive experience that everyone seemed to be on the right side of open science. As soon as I was saying I want to pre-register that, I got a lot of goodwill. The other kind of benefit of doing my discovery sample was that I now had a sense of how big my atypical class was going to be, which before I started this, I'd wildly overestimated. And so now I knew roughly by what extent I needed to bump up my initial sample size. Downsides, I don't know how many of you have worked on manuscripts with other people yet, but personally I find once you're at the manuscript stage, you get a lot of detailed comments. At the pre-registration stage, not so much. And so maybe some of the things that I would have liked to have been challenged on early didn't get challenged. So my lesson from that is to specifically ask for detailed feedback at that stage. The other the big issue for me was that the protocols, although we'd all set out to align our protocols from the outset, it didn't actually work out quite as neatly as we'd hoped. It turned out in this case that one study had actually used a different measure at one of my key time points, and that didn't get picked up until after the pre-registration had been submitted, so that was a great surprise to everyone. So my massive recommendation here is, if you are not using your own data, ask the person who is collecting the data to send you a dummy data set before submitting it. If I'd have just known the variable names, that would have saved me a lot of tears afterwards. It wasn't deal-breaking, because all I've done is I've empirically checked that my measures are comparable, and then I've also checked that when I include and exclude this kind of risky cohort, that none of my main findings change. But still, it wasn't the nice streamlined process that I was hoping for. My next challenge that I set myself was to check whether my latent class analysis converged on similar class profiles. And because I'd been through this once before, I knew I needed to pre-register what I would do if my model fit statistics were in conflict, because that happened first time round. And I knew I would have to operationalize what counted as atypical attention, because in full honesty, when I said, oh yeah, it predicts attentional focus, that was a lucky outcome the first time round. I didn't actually set out to make that my criteria. So I was very strict with myself that was going to be my criteria for atypical attention this time round. And so my recommendation from this is that it's really useful to map out decision trees. Ask someone who doesn't know your exploratory data to look them over. One thing that I didn't include in my pre-registration was what I would do is if I didn't have a clear normative class. Luckily, in my replication sample I did, but when I pulled my data into a superset, it actually wasn't so clear-cut, so I kind of narrowly dodged a bullet there. So any kind of things that aren't going to work out the way that you're hoping will work out, you kind of need to map out and work out what your contingency plan is there. And then kind of the critical point of this study was to run these pre-registered analyses to test whether I would get a predictive association between atypical development of attention and 3-year outcomes. So here my kind of benefit was that my co-author revisions were confined to the text and not to the analyses. So I've had really, really useful feedback on the framing, but no one has tried to get me to add in the extra variable or to do it a completely different way, because it's all pre-registered. So whilst I would advise actively seeking out robust comments on your pre-registration, by the time you're writing it up, you kind of want people to leave you alone. So that was quite a nice benefit of pre-registration. Risks. I did have more missing data than I anticipated. I had a plan for how I was going to deal with that. My plan didn't work out quite as I hoped, because I had even more missing data. So again, I had to use my supplementary materials to kind of declare what the differences were. And I also had, as is often the case in clinical and special populations, very non-normal distributions. And again, I had the plan for how I was going to deal with this, and then the plan didn't work out. So again, had to kind of work out a plan C. So once again, my recommendation is map out decision trees, get someone who's experienced in handling that kind of data to look them over, but also use your supplementary materials. It's not necessarily the end of the world as long as you declare it in a really transparent way. Okay, so then moving to what I actually did and what I actually found. So I was able to draw together 4 cohorts for this replication study. So I had the second batch of infants from BASIS. So these are infants who were not included in my discovery sample. And then I had a cohort from Sweden, Belgium, and Netherlands. That gave me a hefty 412 infants compared with nearly 300 in the discovery sample, so I was fairly optimistic that I was going to capture this atypical development profile accurately. And then I also combined the two in my supplementary materials into a kind of an uber sample, just to check that having a massive sample didn't change any of the key findings, and it doesn't. And then I just ran the latent class analysis approach that I talked you through earlier to see if it would predict 3-year outcomes. So here's what I found. So once again, I'm running the model and adding extra classes until I'm no longer improving model fit. And this time, I get 5 classes instead of 4. And that's not a huge surprise because the bigger your sample, the more classes you are likely to find. So that in itself is not worrying. This is what the classes look like. So to kind of break that down a little bit, we have the bulk of our sample in our normative profile, so that's 64%, and again they're showing that nice clear developmental increase between infancy and toddlerhood, and that's kind of broadly comparable to my 61% in my normative class in my discovery sample. I get again a profile showing just high attentional control across the board from infancy to toddlerhood, and that's 10% versus 17% in my discovery sample. And then I get this really weird one, low focus and high shifting. So they're scoring low on these attentional focus measures and then weirdly high attention shifting. It's a very small subset, and it turned out to be specific to just the Swedish kids. So I don't know what's happening in Sweden that has driven that, but I'm not going to bet my scientific career on it. So I discuss it and I just then leave it over there. It's not particularly predictive of anything, and I don't think it's a very generalizable class. Then I see this low attentional control class, that's 13% of my sample, roughly equivalent to 16% in my discovery set, although you'll note here that they don't quite increase to the same extent. They're not hitting the same values as the normative class in this sample. And then this is the group that I'm really interested in, this plateaued attentional control. So they're not quite showing the dip that they showed in my discovery sample, but they are showing a kind of attenuated increase. So they're not showing the developmental increases that one would expect from infancy to toddlerhood. And then when I use my kind of pre-registered criteria for who's showing atypical development of attentional control, it's these 3 groups, the plateaued attentional control group, the low control group, and the low focus, high shifting groups. They're all showing significantly lower attentional focus at age 3 compared with my normative sample. So those are the groups that I'm then going to do the predictive associations with. So of course that's the big pre-registered question, is this atypical development of control of attention predictive of anything? So here's the big scary table, but I just wanted to kind of orient you to the fact that all of the omnibus tests were significant, and they all survived kind of correction for multiple tests, with the exception of adaptive functioning when I exclude all the missing data. That's more than likely to be because of the power issues, because I had to chuck out a load of infants in that case. So I don't think it changes the findings, but it's just a note of caution. And then to kind of go through each of the measures, what we see is that in terms of SRS, so this is our measure of autism traits, the normative sample, as you would expect, quite low, but the plateaued attention development profile is showing significantly higher SRS scores, so higher autism traits, and that's within the realm of clinical concern. The low attentional control class also showed significantly higher SRS scores than the normative group, but they don't quite hit the levels of clinical concern that the plateau group do. And in terms of ADHD traits on the CBCL, we see low scores in the normative class and significantly higher scores amongst the low attentional control profile. Again, not clinically significant, but these are 3-year-olds, so it's a little bit early to be expecting that. We also see significantly higher ADHD traits amongst the plateaued attention profile, but to a slightly lesser extent. And then in terms of adaptive functioning, So our normative class is actually slightly lower than in the discovery sample, but nevertheless they are significantly higher than the plateaued attention development class. So we're seeing adaptive function difficulties amongst the plateaued attention development class and amongst the low attentional control class. So then I kind of just did a little bit of exploratory analysis to test whether we see any group differences. Infants with a family history of autism who are at elevated likelihood of autism weren't more likely than typical likelihood infants to be classified to either of the atypical attention control classes. But children who received a diagnosis of autism were 4 times more likely to be classified to that plateaued attention development class compared with infants without a diagnosis. Nevertheless, I think this is really important. The majority of infants who got a diagnosis of autism were in the normative class. So it's certainly not the case that attentional control is disrupted in infancy for all infants who get a diagnosis of autism. But it does seem to be a real issue for a small minority. And there was no significant association between autism diagnosis and membership of the other classes. So then, kind of bringing this together, what can I conclude from this? Well, my first conclusion is that there's robust evidence for the heterogeneity of development of attentional control amongst infants with an elevated likelihood of autism. What I didn't show you was that my data-driven attentional profiles were explaining more variance in my 3-year outcomes than the single time point measures do alone. Only a minority of the autistic infants show atypical attentional control. And so in combination, that may well explain the mixed findings in the literature to date, that we've been overlooking this heterogeneity. And so what I think people should take away from this is that research and screening programs should be using data collection and analysis methods that take into account this heterogeneity up front. My second conclusion is that attentional control does indeed seem to be a transdiagnostic system implicated in autism and ADHD. So it could be that this is a risk factor, perhaps interacting with variation in other systems. So I'm not saying it's causal in and of itself, but perhaps in interaction with atypicalities in sensory or social processing in autism, or with reward processing in ADHD, it might be the thing that's giving rise to behaviours that meet thresholds for clinical concern. But interestingly, autism and ADHD seem to be showing slightly dissociable profiles of atypical attention development. So that low attentional control profile, where we see low scores early in infancy, was more predictive of high ADHD traits and not so much autistic traits, whereas later disruption seemed to be predictive of the autism profile. So it could be that actually different aspects of the executive attention network or networks are being disrupted here, and I think that would be worth following up in kind of neural studies. But the other possibility is that our early parent report measures are sensitive to autistic traits like monotropism. So there's some interesting work by an autistic scholar called Dinah Murray, who's suggested that autistic people are very monotropic and that they get into these attentional tunnels. And once you're in an attentional tunnel, you find it very difficult to get out of it. And so that would mean that when we're asking parents things like, does your baby stare for 5 to 10 minutes at a single toy? That could be evidence of a monotropic style, that they're not necessarily using top-down control there. So that would be more likely to be picked up in those early measures than in the toddler measures, where we're also asking things about distractibility and persistence. So that's something that I think we need to really kind of unpick, perhaps using experimental and behavioural measures of attentional control. So we definitely need to validate this approach with other measures. But in the meantime, I think that it might be worth kind of pursuing studies on genetic polymorphisms that are associated with atypical attentional control rather than the broad diagnostic criteria of autism per se. And then kind of the big thing that I was setting out to test is that, yes, atypical development of attentional control is associated with elevated autism traits, elevated ADHD traits, and lower adaptive functioning. And so, of course, we need to follow up in later childhood to see if these associations remain, particularly with ADHD, because 3 years is too young to be diagnosing ADHD. But it does seem to indicate the need for early screening amongst infants with an elevated likelihood of autism to identify that subgroup who might need the targeted interventions and extra support to cope with the day-to-day attentional demands of life. And then in terms of kind of my reflections on the pre-registration process, so It's certainly possible to pre-register data-driven analyses. I was worried that I was going to be setting myself up for a failure, but I would definitely, if you're going to try to pre-register something data-driven, I would recommend running it on a discovery sample first. I would certainly recommend mapping out decision trees for every step of the process and getting people to scrutinise those decision trees. But not get disheartened if you don't pick up everything. You will miss some things, so just use your supplementary materials to declare those. And then, I can't stress this enough, if you're using other people's data, get them to send you some samples so that you know that you're working with the variables that you set out to work with. So I need to thank all of the families who contributed to this study, both from the BASIS and the EuroSIBS studies. My supervisors, Tony Charman and Emily Jones, but also the BASIS team who collected this huge raft of data, and the EuroSIBS teams in Sweden, Nijmegen and Ghent. Our project funders, MRC, Horizon UK and EU AIMS, and then my funders for the last year, ESRC, and my current funders, University College Oxford. And thank you for listening.
Thank you. Do you have any questions? No? No one's shy? I'll go on you.
Last question, yeah. Might be more of a comment, or maybe I'll try and put a question in somewhere. It was really interesting to hear, in general, your approach to this. I've used LCA and seen lots of use of LCA, and I generally find the sort of subjectivity in sort of what people find be a concern. And so I think it's really nice to see this being successful. So one of the thoughts I was listening was when we're sort of teaching LCA, normally we say, well, you've got your model fit statistics, but also you should also, in equal measure, be taking into account what the classes mean in the interpretation. And so, and if not, then we'd be calling this something like unsupervised learning, because like our machine learning has something. So there's a sort of, sometimes there's a challenge of replicating subjectivity and sort of applying, I guess maybe that sort of idea of decision trees could be applied there, but when you do it again with a different sample, how can you apply exactly the same metrics to label those classes and to characterize those classes? And so that made me think sort of also maybe, and also what you're doing here is you're replicating two things. One is the formation of the classes, and the other is how well those perform. And so another thought was, well, maybe an even sort of harsher test would be, don't redo your classes a second time, but just keep exactly the same class structure that you had in your discovery sample and test the predictive performance of that in your replication sample. In other words, so then you're saying, do these classes work in predicting some outcome? But what you've done is a very nice hybrid of doing both. You're essentially replicating, can we do both the measurement part and the prediction part? So I don't think there was a question in there.
Yeah, I think there's a middle step that I did as well, though. So I pre-registered the idea that I would define how I was describing those classes on the basis of that 3-year attentional focus score. So that wasn't in the latent class analysis. That was like my objective measure of how I would label them. But I think it would be really interesting to see if I could ascribe a top-down criteria for attentional focus classes. But that's not what I wanted to do here. I wanted to challenge the way it's normally done in my literature, which is everyone already has these preconceived notions about what good and bad attention is. I wanted to see if we took those notions aside, what would come out.
I have a question. How did you find out where to ask for data from other people?
Yeah, okay, so I glossed over that because it was super easy for me. I'm part of the BASIS study, and so that in itself is part of the EuroSIBS consortium. So it was all laid out nice and neatly for me, so I have no useful advice to give you.
Okay.
Try and shoehorn yourself into a big study, I guess, would be my advice.
Any other questions? One other question is, there's, following on from the replication crisis, as it's called now, is the sort of measurement crisis that's sort of waiting to hit everybody. And the idea being that the measures that we use concern is whether you have any comment on that, whether you, in your reading and collecting, think that there was an issue with measures and outcomes from those measures are used inconsistently, which is tends to be my finding in ADHD, that it is a very pick and choose, whatever you like, based on convention rather than any justified reason. But I don't know.
Yeah, okay, so I guess I'll tackle that in two parts. So I had two sets of measures that I needed to worry about. I had my attention measures and then I had my outcome measures. In terms of attention, so there are these very dominant measures of attention in the field, and there's a lot of good things going for that because there's a lot of literature from the typical development field showing us how these things perform in the wild. And that's great when you're interested in clinical populations, because a lot of the time when I'm looking at other stuff, I'm trying to make up my measures as I go, and that's horrible because you don't know what you're expecting. But I guess the flip side is because they were not developed with autism in mind, you get these issues like they're potentially sensitive to monotropism. So yeah, it's a kind of double-edged sword, I guess. You know, I obviously looked at kind of internal consistency of my scales and they're performing nicely, and I had no concerns on the conventional thresholds for how we would establish whether these measures are working. But yeah, I don't think there is enough scrutiny on whether they're working in the same way in clinical populations. That's an important piece of work that needs to be done. In terms of my outcome measures, again, because I was pre-registering it, I had to say up front what measures I was using, and there was this kind of, do we go for the objective measures because then we're doing something different from parent report, or do we go with parent report measures and potentially pick up like a negative halo effect? Because I wanted to study ADHD traits, and there aren't any robust measures of ADHD for 3-year-olds. I kind of had to go with parent report. So then that kind of took me down a certain pathway that I had to then match my autism measures to that. So then you're like looking at, well, what's the best or most accepted autism parent report measure for 3-year-olds? So yeah, you go down a forking path in a different way, I guess.
Yes.
Yeah.
So yeah, so in a way, so long as you're transparent in doing that as well.
Yeah.
Is obviously key.
Yeah, I mean, a useful next step would be to see, is this predicting ADOS scores, for
Yeah,
example?
yeah.
I guess by looking at how it relates to diagnostic criteria, I'm slightly doing that, but the whole point is I'm not trying to say I'm describing autism. I'm trying to describe atypical attention control. Yeah.
Yeah. I mean, have you heard of the CRUD factor?
No.
The CRUD factor, there was this philosopher of science called Paul Meal, and he came up with the CRUD factor, which is basically everything correlates with everything.
Right.
And nobody knows exactly how much of variance you can explain by this CRUD factor, but people say that it's like a correlation, Pearson's correlation of 0.3, which is usually what's reported across psychology as a good correlation. And it really undermines the idea that things can be correlated with different things. But what we are just looking at is the fact that things just generally correlate. And this is just like, I can see the car crash waves, happening in slow motion. This is going to happen where most of our measures that seem to have correlated don't actually correlate. But I don't know if that was right.
Yeah.
It's interesting. Right, is there any other questions that we can move on to tea and coffee? No? Okay, well thank you very much.
Thank you.
Lovely talk. So yes, it's tea, coffee, biscuits outside. Grab what you like, what you do. The one that desperately needs attention.
Yeah.
That's where it matters. You're just one step away from the clinical setting. So yeah, it's really great. Nice one. Do you wish your post certificate?
Amy's post.
No, that will stay.
transferring. Right, let me plug it in. Oh, sorry. You know how and then halfway through it's like, hyphenating. Double that, but don't worry, get this maybe self. No, I'm fine.
That's not yours, right?
No.
So we have these two. Which one would be?
This one, isn't it? Yeah. And then you just change input. Okay, it should be painless.
It will, don't worry. We've done it before.
That was good.
I'm getting used to different computers.
Yeah, that's good. At least it's not a Mac, eh? So I have started on it. It's when they're rolled sometimes.
Is this okay? Do we don't need to do it?
If it works, it works. Right, maybe I should. Oh, I'm not paying to the internet. Oh, when I'm in slideshow mode, I don't need to do anyway.